Hi everyone. Today we are going to see the topic industrial fermentation technology. From this, we are seeing the ethanol production. These are all some of the microbes producing ethanol. There are a spectrum of microbes are available for producing ethanol, but we have given only a minimum number of microbes which are giving a viable result. For in yeast and bacteria, there are three species in yeast. One is a Saccharomyces cerevisiae, another one is Saccharomyces ellipsoideus. Third one is Cluveromyces fragiles. For these yeast media will be the starch and sugar for all the three yeast microbes. Then second one is bacteria, the Zymomonas mobiles and the Candida pseudotrophils, and third one is Candida utilis. For this starch is the medium, whereas Candida pseudotrophils, lactose and whey, whereas third one, that is sulfide and base of liquor. These microbes can be used for the mass production of ethanol in industry. So this is the biosynthesis of ethanol. So these are all the steps involved in the synthesis of ethanol. This is a starch, base and cheap raw material. On hydrolysis, it yields glucose. As we all know that glucose undergoes glycolysis, it provides pyruvate. That process, pyruvate dehydrogenase will act on the pyruvate and it releases the CO2. The presence of thiamine, pyrophosphate and magnesium ions. Then it produces acetaldehyde. Then alcohol dehydrogenase enzyme along with that NADH, it produces ethanol. So these are all the chemical equation. 100 molecules of 100 gram of glucose molecule will give you the 51.11 gram of ethanol along with 48.89 gram of carbon dioxide. It is an exothermic reaction. During the reaction, the 28.7 kilocalorie of energy is produced. So in the fermentation process, the temperature will keep on rising, so that the cooling agent or cooling devices which has to be used to maintain the temperature between 21 and 26. The maximum it goes up to 27 degrees Celsius, and go beyond that, the fermentation process will be stopped. It is entirely an anaerobic process. Aerobic process also gives the development of microbes very successfully. The colony, the number of cells will increase, but the production of ethanol will not be possible. So when there is an absence of oxygen, it undergoes an anaerobic process. In that anaerobic process only the ethanol will be produced. So ethanol production by fermentation. So these all the steps are involved in the industrial production of ethanol. So the raw materials may be carbohydrate sources like molasses, corn waste, wheat waste. These are all some of the carbon sources. So that raw material has to undergo pre-treatment and sterilization procedures. Sterilization may be heat sterilization or chemical sterilization or autoclave, whatever the process. Then this is a pre-culture. Pre-culture is a inoculum. Our desired microbes identified, isolated, and made into pure culture. That pure culture taken is a inoculum, that is a pre-culture. Then a fermentation. The fermentation process, as we discussed earlier, the biosynthesis of ethanol, that is a fermentation process. Then separation from the fermented material. We have to separate the broth and hard materials. That separated broth then underwent distillation for frequent continuous heating, and all the volatile materials are get evaporated. with the help of condenser. Again, it turns on to liquid state, through which we can separate the ethanol and other materials. Then dehydration, the removal of water, so that we will get absolute alcohol. Then denaturation and stillage. Stillage are the waste substance of the fermented process that may be used for manure or some food for some cattle, and like that we can use. For the protection of the ethanol, we can use sugarcane molasses. It contains 10 to 18% of carbon source. The pH will be 4 to 5, approximately 4 to 5, but exactly it has to be 4.5. For nitrogen source, we add ammonium salt. The inoculum, quantity of the inoculum is depends on the fermenter size and the volume of the nutrient media in which we are kept in the fermentation chamber. Usually there are 4% of inoculum is used. For example, 100 liter capacity of the fermenter, we used 4 liter of inoculum for the process. The maximum fermenter size will be 120,000 gallons. Initially, for the growth of the microbes, we supply the oxygen. If I getting the oxygen, it will grow fast and it will make a multi number of colonies. Then the growth will be very faster. Later only we have to reduce the supply of oxygen so that it started to produce ethanol. Normally this process takes around 48 hours to 72 hours. Every microbes has its own doubling time. From that also we can calculate how much colony will be produced and the cells are produced. So this is the stages of fermentation methods of production of ethanol. So the uses of ethanol. So there are so many types of alcoholic beverages and drinks, then medicine productions, and they act as a solvents. Then number of cosmetics production, and most of the all it is a renewable energy source. It can be used as a fuel for the automobiles. So benzene plus ethanol, it is called a gas oil. Then a diesel plus ethanol, it is diesel. So the 40% of ethanol can be used along with the fossil fuel for automobiles, and it is also serve as a disinfectant. So these are all some of the applications of the ethanol.